Hello, 30, and welcome to the video on DNA replication. We're going to start today by talking about what is the importance of this. We're learning about the S phase. So you learned in the cell cycle that there's a couple different phases, one of them being the S phase or replication. So what we're going to finish talking about today is how this S phase actually creates a copy of the DNA through the process that we're going to talk about. You know you've successfully completed and are starting to understand this if you can identify the words helicase, DNA polymerase, ligase, and free nucleotides. You should be able to explain the process of replication, telling me what is the role of helicase, how does the diagram work with DNA polymerase, explaining Okazaki fragments, leading strand, lagging strand, and ligase. And lastly, why is replication super important to the cell and further processes that it's going to go through. So we're going to start today with homologous chromosomes. All right. So in your notes, you should be able to write these down as we go. We have two sets of DNA, one from our mom and one from our dad. Okay. Homologous chromosomes are similar in that they have the same genes that are the same size, but they're created with variation. So for example, when we talk about eye color, if you have brown eyes, you can have the gene for brown eyes, but you can also have the gene for blue eyes because brown is dominant to blue. So one of your chromosomes can hold the big B gene, which would represent brown, and one of your chromosomes would hold the little b gene, which is essentially for blue eyes. So you can have homologous chromosomes, same genes, same coding, but they're just showing different variation. Humans have 23 pairs of chromosomes, or 23 homologous chromosomes. And the number of chromosomes in our body are called our ploidy. All right, so then remember yesterday's video had diploid, haploid, polyploid. Ploidy just means the number of sets of chromosomes. So here is now where it's going to get a little bit sticky. I'm going to go through what is DNA replication, and we're going to fill out all the parts that tell you what each piece is for. And then what I would like to do is go through the picture. So at one point, all of us was a big cell. Now we have trillions of cells, all right? So we know this happens through the process of DNA replication. So what we need to know about in DNA replication are a couple key players. There's helicase, which is an enzyme, because it ends in an ASE, that unwinds and unzips the DNA molecule. We also have DNA polymerase. Now, DNA polymerase actually has a couple of different jobs. It adds free nucleotides. So reminding yourself a nucleotide is those A, T's, C's, and G's, okay? So it's the backbone part and the A's, T's, C's, and G's. It proofreads when it's done to make sure there's no mistakes, and then it fixes any mistakes it comes across. So that's DNA polymerase's job. Then we have ligase, and ligase is glue. Again, another enzyme, but essentially it glues DNA back together. Now that's going to make more sense when we start walking through the process. And then last but not least, we have free nucleotides, which we get from the food we eat. So here's all the players. There's not very many of them, but what you're going to find out is this process is long. So please follow along in the video, pause as needed, or rewatch if you need to. So here is our double helix strand of DNA. Okay, so this is what we would call complementary strands. Here's my first strand, up and down, up and down. Here's my second strand, down and up, down and up. And these are complementary to each other, right? They're not identical. They're complementing. So A's are paired with T's, and C's are paired with G's. So the very first step, which you have in your notes already, is helicase unwinds and unzips the DNA. So you have this picture, but now you just need to add the arrow and what helicase is. The next step is something that looks like this. So, I gotta move this. What you are seeing now is you have a picture that shows, I went too far, guys, sorry. You have a picture that shows this. All right. So what you need to do is you need to add helicase's name onto your sheet. You need to add the 5, the 3, the 5, and the 3. And that's going to become important here in a second that I'm going to explain. What I would also like you to do is once you have all of this, you're going to add DNA polymerase. That's this guy right here, and you're going to show the arrow this way. You're still on the second picture. And what you're going to do or attempt to do is try and show this process. And I'm going to walk you through that process. So what I would recommend is in your second picture, this is what you draw. So everything you see here is what you draw. So what did we just look at? Well, let's go back and talk about it. So this is the start of DNA replication. Helicase unwinds and unzips the DNA. So there it is, it's being pulled apart from each other. The complementary strands are being unwound. Now, helicase is going to do this all the way through. Now, what its main job is to unwind is so that DNA polymerase can bind on the 5'. We call this the 5' side. Okay. Now, the reason it's called 5' is the way that the sugar is set up with the phosphate. You don't need to know that. You just need to know that this is a 5' side. DNA polymerase can only read in the direction of 5' to 3'. Okay, so it reads 5' to 3' in one big swoop. So you'll notice as it's reading, it just adds complementary nucleotides to this. So if this was an A, this would be a T, this is a G, this would be a C. So it's having no issues reading. What ends up happening is there is a problem on the bottom strand. This bottom strand is actually called the lagging strand, and we'll label all this in a minute. This bottom strand has the issue because the 5' is down here and the 3' is over here. I just finished saying that polymerase can only read in a 5' to 3' direction, which makes it a problem because unfortunately it has to read it kind of backwards. So what happens is there's a primer set out, kind of like a little start sign for DNA polymerase, and it binds to that nucleotide. And what it does is it reads it 5' to 3'. So it reads it in the opposite direction. Once it gets to the end, this little guy is going to jump all the way over here and read it again. So here it is again. It's jumped, and now it's going to read it in that direction. This process is going to continue down all the way through the DNA. So the top strand is being read with no issues, and that is called the leading strand. The bottom strand, however, has these little breaks and holes, which is called the lagging strand. So this, ladies and gentlemen, is what I would draw in picture number 3 that you have, showing that the leading strand was perfectly copied and that the lagging strand has these little sections of DNA that are not combined together. Those sections, and what I would do is I would just write yourself a note off to the side or label them. They're called Okazaki fragments. The reason they're called Okazaki fragments is because of who discovered them. A man, I believe it was a wife and a husband team. Anyways, so they discovered the Okazaki fragments and essentially saying, okay, this lagging strand has to be read 5' to 3'. And since it reads and then jumps backwards, reads and jumps backwards, it leaves these little holes. And that's why they're called Okazaki fragments, because they discovered those holes. So how do we fix these holes? Well, this is what your next picture looks like. So picture 3 looked like this, with your DNA polymerase still labeled. DNA polymerase here, helicase, all your sections of Okazaki fragments, and one perfect leading strand. Your very last picture, or your picture 4 here, okay, is going to look something like this. So the Okazaki fragments are on the bottom, and you have your full complementary strand for your leading strand up here. So what you can do, and you don't have to write all of this in, you can just write a little bit of it in, or you can copy the whole thing. You have 2 original parent strands, or the original copies. They're complementary to each other. You'll notice this A paired with this T paired with this A, and so forth and so forth. What is going to happen is now that they're broken apart, the red strands are going to become now its complementary strand. So now this original parent strand down here is identical to the new strand made up here. If you compare them, they're identical. The reason is because they're both complementary to this strand. This bottom strand here is complementary to this original parent strand, but it is identical to the leading strand up here. All right, so you can see how we can replicate DNA. By having complementary strands, you just need to create the other strand that matches it. The issue is this bottom strand is filled with Okazaki fragments. So what has to happen is DNA polymerase is going to read and fix the mistakes on the top strands. As it's doing that, ligase is going to go along and it's going to glue together any issues, and DNA polymerase is going to read it. So I'm going to go back through this to tell you what to draw. So when you are drawing this, you've got the Okazaki fragments. You don't have to label them. But in your drawing, you have two strands that you've created. What I would like you to do is just draw in the DNA polymerase on the top and the ligase on the bottom. All right. And whether or not you want to draw ligase over and over again like this is doing, that's fine. It's up to you. Or you can just draw it once and then make yourself a note. Ligase will glue the Okazaki fragments together. And then I would make yourself a second note underneath this and say DNA polymerase reads both strands. So it reads both sets of complementary DNA. Because DNA polymerase is trying to make sure that there's no mistakes, no issues, okay? So that's kind of what the role of replication is. So if we were to do, when we do our drawing quiz, because we will do a drawing quiz on this, and it's going to look a little different than this because it's going to be a little bit harder to do. I'm just looking for you to explain to me what is the role of each piece and how does it work, all right? Drawing wise, this may have been a bit confusing. So just go back and draw what you think is the best for each step that you can understand. So if that means that the stuff I told you to draw, you're like, I'd rather draw this, as long as your understanding is there, that's the key point. So again, this is what I will give you, and we will talk about it and we will review it. Hopefully this kind of makes sense. I will post another video to kind of explain DNA replication without a drawing so that you can see what it is as a process instead of just drawing it. And then we can talk about it together later this week. Have a great day.